All right, let's jump right into this explainer. If you've ever looked at a cell diagram and just saw a bunch of, static floating blobs, well, prepare to have your mind changed. Today, we're turning that textbook image into a bustling, dynamic city. It's actually wild to think about, but as you're sitting here listening to this, you literally have over 30 trillion of these complex automated cities operating inside of you, right now. We're going to explore the incredibly precise machinery that keeps you alive, molecule by molecule. So here's our roadmap for the journey. We're taking a full tour of this microscopic metropolis. We'll start at the outer border, venture inside to the busy factory floor, visit the highly secure command center, and then we'll watch how this city actively lives, divides, and even perfectly schedules its own destruction. Part 1. Crossing the cell border. We're starting right at the perimeter to see exactly how our microscopic city manages the constant, heavy flow of imports and exports. What's really fascinating here is the stark contrast between pushing materials out and pulling vital resources in. Exocytosis? That's the cell's export business. It's how your body physically releases essential things it's manufactured, like, say, insulin moving into your bloodstream, or sweat onto your skin. On the flip side, we have endocytosis, the active import business. This is how the cell pulls in vital sugars, large proteins, or even traps foreign bacteria that your immune system needs to deal with. And when it comes to pulling things in, the cell actually acts a bit, well, human. First up is phagocytosis, which literally translates to cell eating. A perfect real-world example of this, your white blood cells. They act like these tiny cellular security guards, patrolling the city and physically eating invading bacteria to keep you from getting sick. Then there's penocytosis, or cell drinking, where the cell just takes these big gulps of extracellular fluid. And finally, receptor-mediated endocytosis. This one is highly specific. A substance has to find the exact matching receptor lock on the surface, and only that precise binding triggers the cell to open the gates and bring it inside. Kind of like a VIP keycard. But hey, border control isn't just about those big bulk packages. It's incredibly strict about individual ions too. And this specific turnstile, the sodium-potassium pump, is absolutely mind-blowing. It's found in every single cell in your body. It actively uses ATP, which is the cell's energy currency, to pump sodium out while simultaneously pulling potassium in. And here's the crazy part. The single mechanism is so vital for things like nerve function that it consumes up to 1/3 of your body's entire daily energy supply. Yeah, 1/3. Just to keep these tiny cellular turnstiles spinning around the clock. Moving on to part 2, inside the cell factory. Let's step right past that border and onto the bustling factory floor, better known as the cytoplasm. Now, the cytoplasm isn't just empty space. It's quite literally everything inside the cell except the nucleus. To bring our city metaphor back, think of the cytosol as the atmosphere in the crowded streets. It's this watery, syrupy fluid filling up the space. Then we've got inclusions, which are basically your storage warehouses holding on to materials like glycogen, melanin, or proteins until they're actually needed. And finally, the organelles. These are the specialized buildings and factories inside the city that do the real heavy lifting. Thinking of these membrane-bound little organs with easy, functional names is honestly the absolute best way to grasp how the factory actually works. Imagine an essential protein being ordered by the body. The rough ER, which is covered in these tiny builders, acts as the manufacturing floor to construct the protein. Then, that raw protein gets shipped straight over to the Golgi apparatus, which is basically our cellular FedEx. The Golgi modifies it, packages it up, and ships it perfectly to where the body needs it. We also have the smooth ER acting as a fat and detox factory, lysosomes functioning as the cellular trash cans to break down waste, and of course, the famous mitochondria, the power plant converting food energy into that ATP we talked about a minute ago. Now, while those previous workers had walls, our structural workers are out in the open, but they are just as critical. Ribosomes are those tiny protein builders I just mentioned. Then there's the cytoskeleton. And it's amazing to think that the cytoskeleton isn't just static scaffolding. It's a dynamic, shifting highway system that literally moves things around the cell and gives it its shape. Centrosomes and centrioles? They're highly specialized helpers that only step up when it's time to divide. Then we have exterior features like cilia, these moving hairs sweeping away debris, flagella, which act like a whip to move sperm cells, and microvilli, which are like tiny absorption fingers, maximizing the surface area to soak up all those nutrients. Part 3, the nucleus command center. Let's move inward and see how all of this builds into the highly secure core of the cell. This right here is where the ultimate instruction manual, the DNA, is kept completely locked down behind a protective wall called the nuclear envelope. That wall has specific doors, or nuclear pores, letting only authorized material in and out. Deep inside sits the nucleolus, a specialized zone building parts for our ribosomes. But the state of that DNA actually changes depending on what the cell is doing. Think of chromatin like a massive, unspooled pile of yarn. It's kind of messy, sure, but it's really easy for the cell to read and access the instructions for its daily jobs. But when it's time for the cell to pack up and divide, it knits that loose yarn into tight, super manageable packages called chromosomes, ensuring absolutely nothing gets lost during the big move. Part 4. The active cell cycle. Now that we've got the anatomy down, let's shift our pacing a bit and watch how the cell actually lives, grows, and prepares to clone itself over time. So most of a cell's life is spent in what we call interphase. It's really easy to mistakenly think of interphase as just a waiting room for cell division, but actually, scratch that. This is where the cell spends about 90% of its entire life. It's the main event. During interphase, the cell follows a strict timeline of doing its job, growing, meticulously copying its DNA, and performing these rigorous final checks across 3 distinct periods: the G1 phase, the S phase, and the G2 phase. The absolute easiest way to remember this timeline is this clever cheat code right here. Lock this into your brain. G1 stands for grow. The newly formed cell does its normal metabolism and regrows to its full size. S stands for synthesize. This is the incredibly delicate moment when the cell specifically copies all its DNA, chromosomes, and organelles. And finally, G2 stands for get ready and check. The cell double-checks everything it just copied and makes the final enzymes needed for division. Grow, synthesize, get ready. Simple, right? But why do we even need that get ready and check phase? Well, without strict G1 and G2 checkpoints, a cell might divide before it's perfectly copied, leading to broken, non-functional cells. The cycle controls prevent this. However, not all cells keep dividing. Some enter what's called the G0 state, a resting state where they exit the division cycle completely. This G0 state is exactly why spinal cord injuries are so permanent. Most of your delicate nerve cells enter G0 and simply refuse to ever divide again. They just do their daily jobs indefinitely, never replacing themselves once they're damaged. Part 5, the Great Divide, also known as mitosis. We've reached the climax of our explainer, the dramatic physical cloning process. Now, before we physically split the cell, look closely at these sister chromatids. Because the DNA was copied back in the S phase, we now have perfect, identical twins of genetic material. These sister chromatids are locked together incredibly tightly at the center by a structure called a centromere. They sit there like 2 sprinters at the starting line, wound up and full of tension, just waiting for the chemical gunshot to be aggressively pulled apart to opposite sides of the room. This sequence is a beautifully choreographed dance of division known as PMAT: prophase, metaphase, anaphase, and telophase. Let's walk through it real quick. In prophase, the secure nucleus completely breaks down and the tightly packed chromosomes step out and attach to a spindle. In metaphase, notice how they march forward and line up perfectly along the equator, or the metaphase plate of the cell. Then, in anaphase, the real action happens. Those sister chromatids are rapidly ripped apart and dragged to opposite poles of the cell. Finally, in telophase, the separated chromosomes arrive at the edges and new nuclear walls are quickly rebuilt around them. But wait, having 2 nuclei inside one joint cell isn't the end of the story. Right after telophase, cytokinesis kicks in. While mitosis separated the DNA, cytokinesis is the event where the outer membrane begins to pinch inward, creating a cleavage furrow. Imagine pulling the drawstrings on a pair of sweatpants tighter and tighter until the entire factory is pinched completely in half. The cytoplasm is divided, and what was once one single cell officially becomes 2 totally distinct, fully functioning cells. Part 6, cell death and destruction. To wrap up our cellular journey, we absolutely have to look at what happens when the factory finally shuts down. Because nothing in the human body lasts forever, right? The cell is actually programmed to handle its own demise. I was super fascinated by the stark line drawn here between a cell simply recycling its trash and triggering total self-destruction. Autophagy literally means the cell eats parts of itself. It systematically breaks down its own damaged organelles or internal waste so the rest of the cell can survive and keep functioning cleanly. But autolysis? That is a much more drastic terminal event. This is when the cell deliberately ruptures its own lysosomes, flooding the factory floor with digestive enzymes to break down absolutely everything. The cell actively kills itself entirely. And we leave you with this profound concept: apoptosis, the ultimate biological term for perfectly programmed cell death. Think about that for a second. The death of a cell isn't always an accident, an infection, or some disease. In many cases, it is written directly into the very instructions of the DNA. And this is vital. When apoptosis fails and cells refuse to die, they divide uncontrollably, which is exactly what cancer is. Apoptosis is what keeps us healthy. It proves that in our microscopic city, from the careful imports at the border to the frantic copying in the nucleus, even death is a perfectly scheduled event that keeps the entire human body alive. So what other microscopic miracles are happening inside you right now? Thanks for taking this incredible cellular tour with me.